Hi, my name is Amanda Jerng, and I'll be presenting on disulfidptosis, which is a novel form of regulated cell death and a promising pathway in the development of cancer therapy. I'm presenting from Sugar Land, Texas, and my mentor's name is Natalia Kalatian. I first became interested in this topic as I've always been attracted to cell death, like apoptosis, and even more interested after I realized it could be used to treat real cancer patients. So what is disulfidptosis? It's a strange name, but it's a form of regulated cell death caused by intracellular accumulation of cysteine and disulfide stress. I've broken it into three parts, which are glucose starvation, NADPH depletion, and actin cytoskeleton collapse. So why regulated cell death? Why is it becoming popular as a form of therapy? And to answer this question, I'll be asking another question, which is why targeted therapy? Targeted therapy is the use of small molecule drugs to target specific metabolic vulnerabilities in cancer, causing cell death and increased efficacy of existing treatments, as well as reduced drug resistance. Specific alterations in cancer arise due to the need to meet increased biosynthetic and bioenergetic demands associated with uncontrolled proliferation. The first is reprogramming of metabolic pathways, and this includes the Warburg effect. The Warburg effect is essentially where cancer cells will increase production through increase activity through aerobic glycolysis, regardless of the presence of oxygen. De novo synthesis pathways are also upregulated or deregulated. Two is the upregulation of nutrient transporters in order to meet higher biosynthetic demands in cancer. However, this presents a very large metabolic vulnerability as cancers are highly dependent on specific nutrients, and by inhibiting access to these nutrients, we can cause cell death. Three is reactive oxygen species, or ROS, which are generated naturally through oxidative phosphorylation. So naturally, increase in metabolic activity will increase accumulation of ROS, which can cause widespread cellular damage and cell death. So why regulated cell death? Regulated cell death allows us to induce targeted therapy in a highly controlled and selective manner. The amino acid cysteine is essential for maintaining malignant viability. Cysteine has the ability to form disulfide bonds, and this is essential for protein structure in the form of disulfide bridges, and as well as forming the cysteine dimer, which is essentially two cysteine monomers bonded by a disulfide bond. Cysteine is also critical for antioxidant synthesis. Cysteine is a precursor of the antioxidant glutathione, which is essential for combating ROS, which is increased in cancer cells. Glutathione is also used to inhibit ferroptosis, which is another form of regulated cell death. Compounds such as Avastin and RSL3 have been shown to induce ferroptosis as a form of therapy by inhibiting cysteine transporters. Now that we've established that cysteine is essential for cell growth and viability, we can talk about how do cells maintain an adequate cysteine pool. And this is done predominantly through the transporter SLC7A11, which is it's essential to know that this is a cysteine dimer transporter. This transporter is also upregulated in majority of cancers in order to meet higher need for antioxidant production. Another prominent form of cysteine acquisition is de novo synthesis through the transsulfuration pathway, or the TSS. However, the TSS is generally not adequate to meet the high demands of cysteine in cells. Therefore, cells rely more on the SLC transporter. Some other methods include direct cysteine transporters, so not the dimer, and recycling glutathione and polypeptides within the cell. The SLC transporter imports dimeric cysteine, which must be reduced into its monomer form in order to be used in biosynthetic pathways. The accumulation of dimeric cysteine results in disulfide stress, which is characterized by the increased disulfide bonding in specific proteins, leading to cell death. The intracellular reducing environment responsible for reducing dimeric cysteine is mediated by the TRX and GRX systems, as well as other compounds. The inhibition of these compounds has been proposed as a promising therapeutic mechanism by inducing disulfidptosis. Both the TRX and GRX systems rely on nicotinamide adenine dinucleotide phosphate, or NADPH. As we have previously established, the SLC transporter is upregulated in many cancers, meaning that high volumes of dimeric cysteine are entering the cells. This presents a very large metabolic vulnerability in SLC high expressing cells, as the NADPH required for reducing the cysteine through the TRX and GRX systems is generated by the pentose phosphate pathway, or the PPP. The PPP utilizes intermediates derived from glycolysis. Therefore, SLC high expressing cells are more reliant on glucose. Inhibition of glucose transporters, such as the GLUT1 and 3s by the compounds BAY876 and KL11743, has been shown to induce rapid disulfidptosis. In addition, malic enzymes have been shown to produce equal amounts of NADPH, or have been shown to produce amounts of NADPH equal to or even greater than the PPP, and should be further investigated in relation to this pathway. In the pioneering study of disulfidptosis, it was revealed that cytoskeleton proteins showed the greatest enrichment of cysteine while under glucose starvation. The cytoskeleton proteins, specifically F-actin, also were shown to detach and collapse while under glucose starvation, which implies that the disulfide stress and subsequent disulfide bonding plays a role in cell death through disulfidptosis. However, it's extremely unclear whether the actin collapse itself is the directly results in the cell death. And further studies should be done to determine whether disulfide stress affects other redox-sensitive proteins. Here is a general diagram of my pathway, and thank you for watching.